Okay, so welcome to this next video in which we are discussing heterotrimeric G proteins. So we're currently in the process of discussing the lipid modifications that occur to the G alpha subunits of heterotrimeric G proteins. So we've discussed that most heterotrimeric G protein G alpha subunits get palmitoylated on a cysteine residue that is near the amino terminus. Okay, but some of them, specifically members of the G alpha I naught family of alpha subunits, these also get N-myristoylated. Now, this involves the addition of myristic acid onto the actual amino terminus of the G alpha subunit. Okay, so again, you don't add myristic acid directly onto the protein. Instead, you use myristoyl coenzyme A, which is a myristic acid molecule linked via a phytoester link to a coenzyme A molecule. Okay, so here are these 12 methylene groups of the myristic acid molecule, like so, and then right on the end you then have the methyl group, H3C. Okay, and this is myristoyl CoA. So just like palmitoyl means the acyl group of palmitic acid, myristoyl means the acyl group of myristic acid, this group here. Okay, right. So, how does N-myristoylation occur? Well, basically, it occurs on the first amino acid of the protein. So let's say this is our alpha subunit that is within the I naught family of alpha subunits. Okay, and let's say here is the first amino acid of this polypeptide. So this is the first amino acid. Now, if your first amino acid is a glycine, then N-myristoylation can occur to that glycine. So let me just discuss with you the structure of glycine. Okay, so here is the amino terminus, which is of course free because it's not involved in binding to the carboxylic acid group of the amino acid that was in front of it, because there is no amino acid in front of it. Okay, so then we've got the alpha carbon with a hydrogen coming off it, and the carboxylic acid group, which will be involved in a peptide link to the next amino acid along. Okay, and the R group of a glycine amino acid is that you have just a hydrogen coming off, so it's the simplest of all the amino acids. Okay, so this is glycine. Now, you might be thinking, well, hang on a second, I know something about the first amino acid of proteins. Isn't it always methionine? And I would say you're correct. When you translate a protein initially, you do always add methionine in. So let me just discuss this with you. Okay, so if you have a ribosome here, and it's translating some mRNA, okay, and it's producing some protein here, basically translation begins with the start codon, and let me just extend the mRNA up a bit. So translation begins with the first codon, which is A, U, and then G, like so, which is the one and only start codon. So this is the start codon. And basically, there is a set amino acid which is put in place for that start codon. So the start codon codes for a set amino acid, basically. And that means that since translation always has to begin with a start codon, the first amino acid that you have in your protein is always going to be this set amino acid. And that set amino acid is methionine. Okay, now methionine is therefore often called the initiator amino acid, because when you first produce a protein, it will always be the first amino acid. So hence it's called the initiator amino acid. Now, basically, you often remove the methionine, okay? So even though when you first translate the protein, it is there, often what will happen is you'll cut it off, okay? And that means that the second amino acid in the original polypeptide will ascend to the title of the first amino acid. And if that second amino acid was glycine, then your first amino acid in the polypeptide will become glycine. Okay, and if your first amino acid in the polypeptide is glycine, then N-myrostylation can occur. So this is going to occur for all of the members of the G-alpha I0 family of alpha subunits of heterotrimeric G proteins. Okay, so what's going to happen is the myristoyl group is not going to be added onto a sulfur atom this time because there are none. Instead, it's going to be added onto the amino group to produce an amide link. So, do I have space to do it here? I can have a go. Okay, so in comes our myristoyl coenzyme A molecule, like so. So here's the carbonyl group. Here are the 12 methylene groups. And then finally, the methyl group on the end, CH3. So what's going to happen basically is we're going to cut the bond between the sulfur atom and the carbon atom here. So we're going to cut the thioester link, give one electron back to the sulfur, one back to the carbon. Of course, this isn't an electronic mechanism. This is just to make sense of the reaction. Okay, and again, you can imagine cutting a bond between the nitrogen atom and the hydrogen atom, give one electron back to the nitrogen, one back to the hydrogen, then bind this nitrogen atom here to this carbon atom here. Both of them have 3 electrons in our imaginary flow for this reaction, and then you will bind the hydrogen atom to the sulfur atom. So that isn't, let me stress again, that is not an electronic mechanism. That's just making sense of this reaction, making sure that what's on one side adds up to what's on the other side, and indeed it does. Okay, so you'll regenerate coenzyme A with its original thiol group back again, and now you have this myristoyl group tagged onto the amino group of the first glycine of the polypeptide of the G-alpha subunit. Okay, and that's then called N-myrostylation. Okay, so this occurs for all of the G-alpha I0 family of G-alpha subunits. Okay, so what does that mean? Well, firstly, it means that the 2 that didn't get palmitoylated, which remember were the G-alpha T1 and the G-alpha T2, well, they are members of this family, so they'll get myristoylated. So at least they'll get one lipid molecule added on, which will help to anchor them in the inner leaflet of the phospholipid bilayer. So that's good. But it also means that some of them are going to get multiple lipid molecules added on. They'll get a myristoyl group added on, and they'll also get a palmitoyl group added on. Okay, so some that are within this family and which are not the G-alpha T1 and G-alpha T2, those will get both of them added on, and therefore they will have 2 acyl groups dangling off. Okay, right, so that concludes our discussion of the lipid modifications of the G-alpha subunits of heterotrimeric G proteins. We're now going to turn our attention to the lipid modification of the G-gamma subunits of the heterotrimeric G proteins. Okay, so G-gamma gets a rather scarier form of lipid modification occurring to it. It gets prenylated. Okay, so we're now going to talk about the prenylation of the G-gamma subunit. Okay, so I will tell you exactly what prenylation means. It means that we're going to add prenyl groups on, but that's useless because we don't know what a prenyl group is, but we'll come to that. Firstly, I'm going to tell you the condition that you need to obey in order to be prenylated, basically. Okay, so let's say that this is our G-gamma protein, okay? So here is the amino terminus, and here is the carboxylic acid terminus right over here. To be prenylated, you need to have a certain structure at the carboxylic acid terminus of your protein, okay? And proteins that are capable of being prenylated are called CAX-box proteins. And this is because they need to have something called a CAX box at their carboxylic acid terminus. If you don't have a CAX box, you cannot be prenylated. So only proteins that have a CAX box are going to be prenylated. Hence, all of these G-gamma subunits must be in this family of CAX box proteins. Now, what is a CAX box? Well, basically, it's a combination of 4 amino acids that are right on the end of the protein. So the last 4 amino acids in the polypeptide need to obey a certain pattern, basically, which is called the CAX box. So this is the last amino acid, this is the second to last amino acid, third to last, and fourth to last. Basically, the fourth to last amino acid has to be a cysteine amino acid residue. That's the single letter amino acid code for cysteine. C is the single letter amino acid code for cysteine. So the fourth from last amino acid has to be a cysteine. Okay, the next 2 have to be A. Now, lowercase a here does not stand for alanine. If this was an uppercase A, it would stand for alanine. Lowercase a here stands for aliphatic. Okay, and what that demands is that the 2 next amino acids here, the second from last and the third from last, both have to have aliphatic R groups. Okay, so let me give you some examples, the archetypal examples of aliphatic amino acids. So, I should just say what aliphatic means. Aliphatic means not very hydrophilic, so you don't have any polar groups, no alcohol groups, no amino groups, no thiol groups, nothing like that. In addition, it means not aromatic, so you mustn't have aromatic rings, no benzene rings, basically. So, let's have a look at some examples. So, glycine is an example. Now, I'll show you the R groups of these amino acids. I'm not going to draw out the full structure because I haven't got enough space. All I'm going to show is their R groups. Now, the R group of glycine is that you have hydrogen off. So, basically, I'm doing away with all the core structure. I'm not showing the amino group, I'm not showing the alpha carbon with the hydrogen coming off, I'm not showing the carboxylic acid group, but I'm just showing you the R group, the bit that varies between the different amino acids. Okay, so glycine has a hydrogen atom coming off. Okay, next one up is alanine. Okay, so alanine has a methyl group coming off. So this is a carbon with 3 hydrogens coming off. So that's the R group of alanine. Next one up we'll have valine. Okay, so valine then has a carbon, a hydrogen, it has 1 carbon with a hydrogen coming off, and then it has 2 methyl groups coming off this carbon, like so. So this is the structure of valine. That is again a very aliphatic amino acid. No hydrophilic groups here, no polar groups, no aromatic rings. So it's a nice example of an aliphatic amino acid. Then we're getting even better. Leucine and isoleucine come next. Okay, so leucine is basically just a bigger version of valine. So you have a methylene group, and then you have the same group that valine would have. So a carbon with a hydrogen coming off it, and then 2 methyl groups coming off it, like so. And then isoleucine is basically a structural isomer of leucine. Okay, so if you swap one of these methyl groups for one of these hydrogens, so move a methyl group onto this first carbon and then move a hydrogen onto the second carbon, that creates you isoleucine. So we'll have the methyl group now coming off this first carbon, a hydrogen coming off here, 1 methyl group still coming off the second carbon, and then these 2 hydrogens up here. So that's now the structure of isoleucine. Okay, so those are the 5 main examples of aliphatic amino acids. So you can have any of these in these 2 positions here. Okay, and they don't have to be the same. You don't have to have, it's not the case that you pick one and then it has to be here and here. It just has to be an aliphatic amino acid followed by another aliphatic amino acid. And then the final amino acid is coded X. Now, what does X stand for? Well, X isn't the single letter amino acid code for any amino acid. Okay, what X will determine, the amino acid that you have in position X, it will determine which prenyl group you actually get added on. So basically, what I'm going to show you is that there are only 2 prenyl groups. So prenylation isn't that complicated. There are only 2 prenyl groups that cells add on to proteins. There is what's known as a farnesyl group, and I'll show you the structure of this in a moment. And in order to get a farnesyl group added on, that X amino acid has to be a specific amino acid. Well, not a specific one. It has to be one of 3, basically. It has to either be alanine, okay, and we've seen the R group of alanine, it's just a methyl group, or it can be serine. Okay, and the R group of serine is basically a methylene group with an alcohol group coming off. So here's the methylene group, and then you have an alcohol group coming off the side. Or it can be methionine. Okay, so methionine isn't just the initiator amino acid. You can put it in at other places as well. Okay, so methionine's R group is quite complicated. You have two methylene groups, or an ethylene group if you like, followed by a sulfur atom, then with a methyl group off the end. So that's methionine's R group. Okay, so if you have one of these three amino acids in position X, you will then get a farnesyl group added. The other type of prenyl group, again I'll show you the structure of this in a moment, is a geranyl group. Okay, and basically if you want a geranyl group added on to you, you need to have a leucine amino acid at position X. So basically, the amino acid that you put in position X determines which prenyl group you're going to get added on. So both farnesyl and geranyl are examples of prenyl groups. Okay, so we'll call it there for this video. In the next video, we'll discuss the structure of these two prenyl groups and exactly where they're going to be added on. We'll then discuss post-prenylation modifications, and then finally we'll move on to the G protein cycle.